PowerPoint to go through the basics of transcription and translation. Remember that transcription is taking the message from the DNA and creating a molecule of RNA, and then translation uses the RNA to create a protein strand. Transcription takes place in the nucleus, and translation takes place in the cytoplasm. So let's start with transcription in the nucleus. Okay, there's definitely some similarities between transcription and replication where DNA makes DNA. Here we have our double-stranded molecule of DNA. Unlike replication, in transcription, when we make the RNA, we're just going to use one little segment of the DNA here shown as the transcription unit. There's a promoter region at the beginning, and the RNA polymerase comes along and it finds that promoter region and it separates the two strands of DNA, okay, which is what you're seeing down here. And that's called initiation. The RNA polymerase finding the starting point or the promoter region on the DNA and separating the two strands. Now, unlike replication, as we create the new molecule of RNA, we just use one strand as the template. Okay, so here our bottom strand of DNA is the template, the top strand, nothing happens to it at all. And this gets around the problem that the RNA polymerase, which it's an enzyme and its job is to add the A's and the T's and the, not the T's because it's RNA, A and U and G and C. Remember, it only likes to make the new strand 5' to 3' because it's a picky enzyme. And that gets around the problem. We don't have any of the issues with the lagging strand that we had in DNA replication. So the RNA polymerase moves forward along the DNA template. It reads the A and the T and the G and the C and the DNA, creating in the RNA A and U and G and C, and it continues all the way along, forming elongation. If you look down here, you can see that after the DNA winds back up together in that double helix, so we don't create a big bubble like we do in replication. And here's our RNA transcript that's being created in the pink, and the RNA polymerase continues moving along the entire transcription unit until it reaches this red area at the end, which is called the termination. And this is a sequence of A's and T's and G's and C's that the RNA polymerase recognizes as a stop point. And once it reaches that termination, the RNA polymerase releases, the RNA strand releases, and the DNA is directly as it started, and it can be used again for further transcription if you want to make another RNA. So this is the general basic overview of transcription, using the RNA polymerase and a short piece of the DNA to create a sequence of RNA. Now, if we move forward and we look at translation, this is going to take place in the cytoplasm, and it's going to involve the ribosomal RNA, the transfer RNA, and the messenger RNA. So all of them are going to work together to create a protein. Translation occurs, begins with initiation. Okay, so here we have our messenger RNA. Here's the transfer RNA, which contains the amino acid. And the first stage is we have the tRNA binds to the small ribosomal subunit, and then the mRNA comes along and it finds the sequence that is going to match up with this amino acid. Once that occurs, then the large ribosomal subunit comes along and the large and the small ribosomal subunit join, and this creates the end of initiation. We have all of the players together with the large and small ribosomal subunit. The messenger RNA sort of feeds through the middle, and then the tRNA is also on the inside. Once the large and the small ribosomal subunits join, we end up with these two slots inside where the tRNAs can join. The first site is called the P site, and the second one is called the A site. And then the RNA goes in this groove along the top. Okay? So the tRNA comes in the bottom and it matches up with the messenger RNA along the top. So here you can see, here is the messenger RNA AUG, and it matches up with the anticodon UAC, and this amino acid down here would be a methionine. The next three letters for the codon are GUG, which will match up with the anticodon CAC. So this is the next tRNA that's going to come in carrying amino acid number 2. And now we have 2 tRNAs, one in the P site and one in the A site. Now, the inside of the large ribosomal subunit is very acidic, and that catalyzes the formation of a peptide bond, which is a covalent bond that joins between 2 amino acids. So we can see this little link here, amino acid 1 joins with amino acid 2, and then we're going to release the tRNA from that first P binding site. Let's look at that on the next slide as we continue with elongation. Okay, so up here, the first tRNA is released. Here goes the UAC, and now both amino acids are attached to that second tRNA. The second tRNA is going to move from the A site over to the P site, so now this is in the first position, and the third tRNA with the anticodon to match up with the next codon and the mRNA moves into the A site. The same thing is going to occur. A peptide bond will form between amino acids 2 and 3. The tRNA is going to be released from the P slot. The one in the A slot will move over, and then amino acid number 4 will come in. And this process is repeated over and over and over again and is known as elongation. This will continue until a stop codon moves into the A slot, and there is no tRNA that matches up with that stop codon. So it's basically a signal to release. The amino acid chain is released, the 2 ribosomal subunits separate, which you can see over here, the messenger RNA is released, and the process is concluded. So termination, again, is a result of that stop codon moving in, causing everything to release, and then we have our finished amino acid chain and the end of translation.